it's going to be my pleasure to introduce the next session. What we'll be doing is talking through some of the updates from Oxford Nanopore Technologies, and I'll be joined on stage by James Clark, who's VP of Platform Technology, and Stu Reed, who's our VP of Development. We are actually in a safe social distancing bubble. We do share an office, so these are the only 3 people that we're allowed contact with. So don't worry for our, please don't worry for our health and safety. First on stage, we will bring over James. He looks after our platform technology. He's been with the company for 13 years now. Before that, he was at the Hagen Bailey lab. And so there is no greater knowledge source of how the technology works, really, than James. So welcome on stage.
Thank you. Thank you. Really nice to be here. It's quite a small stage by London Calling standards, but we have a massive online audience. So thank you very much for attending, and I hope you're all fit and well. So part of my role today is to talk through the technology and give you an introduction, as we've got so many people online. I think of nanopore sequencing as being quite new. Maybe that's because it's quite different to the existing technologies, or maybe it's because we keep finding new things to do with it. But actually the numbers show we're pretty well established now. So the Minimum Access Program, that started in 2014. Since then we've distributed over 10,000 nanopore sequences in around about 100 countries. And we estimate that there's 1.7 petabases, that's 10 to the 15 bases of sequence information that has been generated on those platforms to date. We've recently passed a landmark 1,000 publications from our platform across a really diverse range of applications. Those include things like outbreak monitoring, plant genomics, and human sequencing, leveraging those long reads, and really novel features of the platform like the direct RNA modifications. There's some examples here just from the last few months from some really top journals. So if a nanopore talk is a bit like taking a flight, then this would be the safety video. I'm going to run through a few of the kind of features, the background to technology, and apologies if you're a regular London caller, if you're a frequent flyer, this won't take too long. So the first thing I want to talk about is the nanopore itself. It's a really exquisite sensor. It has the ability to distinguish really small changes. And to make a measurement through that nanopore, we have to set up the system. So we have an electrode above and below the nanopore, and we insulate that in a membrane so that the only current you see flows through that nanopore. And then we engineer the nanopore so that different sequences in that pore give us different currents. And that means that when we move the DNA through the nanopore, we get a current trace, and that trace we affectionately call a squiggle, and we decode the squiggle to give us our base call. So to do that, we have a motor protein. That motor protein can act on either DNA or RNA, and it moves the strand through the pore at about 400 to 450 bases a second. That motor is different for RNA and DNA. We also have a choice of nanopore. We have the stable R941 release, which has been out for some time now, and we have the R10 series, which is in early access. An important feature of the R10 is that we deliberately engineer the pore to have a greater contribution from more bases, and that makes it better for resolving homopolymers. We've got a few things we can tweak within the system, such as temperature, applied potential, the enzyme cofactors, and we generally tune the system for accuracy. So DNA as double strand will get blocked in the pore and will need to be ejected. Single-stranded DNA will thread through the pore at about 1 million bases a second, so that's far too fast for us to be able to sequence at the moment. So we have to slow it down, and to do that we use the motor protein. And we have to get the motor protein, the helicase, onto your strands of interest, and to do that we have the sequencing adapter. The sequencing adapter contains a number of features. It has a capture leader which helps it thread into the pore to improve the sensitivity, and because the motor is a helicase, and because we've put a helicase in contact with DNA and ATP, that will do what helicases do and run through all your strands. So we've designed a stalling chemistry, and that stalling chemistry means that the helicase won't do anything in solution and only activates when it comes under additional force when it docks to the nanopore. So in order to get our sequencing adapters onto your strands, we can do that in a number of ways. You can either do that via ligation. Our ligation kits typically take around 60 minutes. Or if you're interested in doing it quicker, you can use our rapid kit. And those, as the name suggests, can do this in about 10 minutes using a transposase. But they do cut the DNA to do this. There are also lots of barcoding options, and barcoding is performed at this stage as well. Another feature of the technology is the tether, and our sequencing adapter connects the molecule of interest down to the membrane. That's how the tethering works. We have a hydrophobic anchor, and that means that when these molecules come down to the surface, because the surface is a two-dimensional fluid, the molecules can diffuse around. So we've gone from three-dimensional diffusion down to two-dimensional diffusion, and that gives us an incredible increase in sensitivity. We get about 3 to 4 orders of magnitude in sensitivity from having that as part of our chemistry. So moving on, the great thing about nanopores is that they are a single molecule sensing technique, but that means they only read one molecule at a time. And although a nanopore, once it's read a molecule, can read a fresh molecule, we really need to raid the platform to have a statistically relevant number of molecules to look at. And for that, we have this modular unit called a well. The well performs a number of functions. It has an electrode at the bottom, and it has an isolated fluid volume that allows the nanopores to act independently from each other. So each well has one nanopore, and we spent a lot of time engineering this well design to make the membrane stable, and I'm going to talk more about that later. You can see in the picture with the red and the green, where we've dyed up the membrane material, which is a tri-block. We've dyed that green, and we've dyed the isolated fluid as red. You can see how these fluids look when we set up the system, and the nanopore sits at the pinch point of those kind of two green triangles coming together in the center. Once we've got that modular unit, we can array it, and we can array it to either hundreds of nanopores, thousands, tens of thousands, and that's what you see if you look into a flow cell. There's one other component we need, and that is an ASIC. We have to get the signal from that electrode into our platform. So the ASIC performs a number of functions. It performs data acquisition and control, but it's also got some nifty little features. It can reverse the potential on an individual well, and because the platform's real-time, because you can sequence a portion of the molecule while it's still going through the pore, that means you can do clever things like the adaptive sampling, the read-until methods that Stu and Dan were talking about. It allows us also to unblock unblock and eject DNA so that we can keep channels running. So the last intro slide is on how we go from our signal, our squiggle, to sequence. So the nanopore measurement, it's very simple in concept. You apply a voltage and you measure a current, but there are many things that can contribute to that current. For example, we know that a huge amount of bases contribute to the current. But if we say that all bases in the pore, that's 10 to the 15, contribute to the current, that's a huge amount of combinations that we have to squeeze into a small current. And that might seem like a bit of a problem, but it's actually a benefit in our system, because although you will see them, you'll see 4 to the 15, you will also see each base within that sequence 15 times. So as it moves through the pore, you get to see it 15 times, and that means you get multiple chances to see something, which is really important in a single molecule technique. So we have our squiggle. It's a very rich signal. There's a lot of information in it. But how do we go from that to a base call? Well, we have to decode it. Our decoder uses a recurrent neural network. And for that to work, we have to train it on known sequences. So we have to take squiggles, raw data, and of known sequences, and we have to feed that into our algorithm, and then it learns, it trains itself, so that when we give it a fresh piece of signal, it can give you the most accurate prediction of that sequence. And those training sets can vary for DNA and RNA, and they can vary for different pore types. So the base call has evolved a lot. The raw read accuracy is now at 97 modal. We've got a stable version that's distributed in Minnow with Guppy. We also have lots of research versions, and Stu will talk through those up next. And not only do we make the base caller available, we also make the training methods available so that people can train their own data sets, and that's particularly important for things like modified bases. So very quickly go through some key features. So Nanopore platform is pretty accurate. We've been improving this steadily over the years. As I said, our raw read accuracy is now at 97% modal. The R10.3 chemistry, that's achieving Q50 results on a bacterial test set, and Q50 is one error in every 100,000 bases. Read length is also a really strong feature of the platform. The platform essentially has no drop-off with read length, and that is the accuracy of the first portion of the molecule is the same as the accuracy at the end of the molecule, no matter how long it is. Users can routinely get M50s over 30 kilobases with pretty decent output. But when you start getting to really large samples of DNA, when you're looking for really long read lengths, it becomes tricky because although the platform has no limit, you have to handle the DNA in a very careful way, and that's been generally speaking a community-led approach. They've done some fantastic work, and those protocols are online. They have this whale scale for naming their reads, and as far as I know, the current record is at 2.44 million bases of a human sample. And to put that in context, if we're sequencing at 400 to 450 bases a second, then you could put that strand into a nanopore, that single molecule going through that strand. You could watch it, you could go and watch an entire game of football, you could come back, and it would still be going through that pore. So it just shows what the platform's capable of. But we know that not everyone can get a high output on these ultra-long reads, and that's something that is an active area of research and something that Clive is very interested in, and I'm sure you'll hear more about that in the near future. So the platform, as I said, the nanopore is very, very sensitive. That means that you can see things like base modifications. You don't actually need to chemically modify your sample. The nanopore kind of inherently sees that. It's part of the rich information, and because of that tethering, you don't need to PCR, and you can read native strands. But what you do need is you need a way of training. You need a way of extracting the information from that really rich signal. And we've worked hard on putting those options in Minnow to make them more available. And there are also research tools which we'll talk through later. As far as I know, nanopore sequencing is the only way to sequence RNA directly. It's very similar in principle. We need a different motor. We need a different library preparation. But apart from that, it works in essentially the same way. There are many more modifications in RNA, and so this has been a really strong community-driven approach. There's some great publications out there, and I think it's going to become more and more important in the future. When we have those large arrays, we can achieve very high data output. Mid-ion gridiron flow cells, we get between 20 to 40 gigabases. Our PromethION record is 220 gigabases from a single flow cell. Another way to think about that is in terms of how many reads you get, and particularly useful for cDNA applications. And that would be 200 million reads of a reasonable length of 400 million reads from a single PromethION flow cell. If you don't want to use all of your output on one sample, you can of course barcode, and there are protocols out there for over 2,000 barcodes in a single flow cell, and Rosemary and Stu will talk more about that later. Of course, the platform's very good for time to result. Rapid kits, very quick. Sequencing starts as soon as you add your sample. It's real time. Because the data is written to disk in real time, you can write analysis pipelines to look at the data in real time, and that can give you really high precision detection of sequence in as little as 50 minutes from extracting your DNA. And the other factor is because the platform is portable. When you can't take your sample or simply don't have time to take your sample to a central lab, you can take your sequencer to your sample, and that obviously improves your time to result. So I'm going to very quickly rattle through our sequencing devices. MinION should need no introduction really. First released as part of the MinION, as part of MAP in 2014. Uses the MinION ASIC. That's 2,048 wells spread over 512 channels. It's a USB device. It's very, very accessible. It uses your laptop, and Minnow is installed in your laptop. Base calling is performed on the laptop as well. So it's deliberately priced to be very accessible if people want to give it a try. What we did learn in MAP is that while a USB device onto a laptop is great, people have different laptop specs. The output of the device was growing, and that meant that it put more of a burden on the laptop, and the laptop could be doing other things. So that causes configuration problems, and really we move towards standalone sequencing devices. So Minnow Mark 1C is that. It doesn't require a laptop, and it has integrated compute. It also has a GPU, which accelerates the base calling. So that really is the standalone mobile sequencer. Gridiron, again, standalone device. It can run up to 5 MinION or Flongle flow cells, and it has really powerful GPUs, and the GPUs in the Gridiron can keep up with the highest output flow cell on the most accurate models. So with the other thing we learned during the MinION Access program was the outputs kept going up, and people often didn't need that output. What they wanted was a small disposable flow cell, and Flongle is the answer to that. The Flongle has the ASIC from the MinION, but rather than have it in the flow cell, in the disposable, it's actually in the adapter, and that means we can put all the cheap bits into the flow cell, and we can lower the price of those flow cells. It does come at a cost though. The Flongle has 126 channels, but that can still generate 1 to 2 gigabases of information, which is sufficient for a lot of applications. So there's been a lot of demand for Flongle, and although MinION and PromethION flow cells kind of scale reasonably gracefully, Flongle is really about a high volume product, which means that we need high volume production. So the production of all our flow cells is done at our own factory. That's about 30 minutes down the road from where I'm talking. That factory has been online for about a year now, and it was built from the bottom up with flow cell manufacturing in mind. So we've been stepping through the Flongle process, the wet assembly process, and we've been automating various parts of that. Some of those parts are automated. We're still working on a few more, but we're making good progress. Here you can see a few videos just showing them in action. That robot is the yellow robot arm that is gluing the silicon piece into the Flongle flow cell, and that can perform just shy of 100,000 units a month. And obviously we can duplicate that robot setup if we need more capacity. So looking forward to ramping up the volumes on Flongle over the summer. PromethION is our highest throughput offering. It is a sequencer which is built for on-demand use, so that if you have multiple flow cells running, if there's an empty slot, somebody can come and start a new run, kick off a run without interrupting the runs that are already on the device. So there's no batching. That we think that's really important for flexibility. And the whole device and the software has been built with that in mind, so that users, perhaps running different projects within an institution, could all have log into the box and monitor what they're doing. The PromethION uses a different flow cell. I'm going to talk a fair bit about that in a minute. It is different to the MinION in that it's built on top of the ASIC and it's tighter pitch. That gives us more output. It has 12,000 wells and 3,000 channels, and outputs in the field have been as high as 180 gigabases. So the PromethION comes a couple of hardware options. There's the P48, which can run 48 flow cells at once, and the P24, which can run 24. It also comes with a powerful compute box, which has GPUs to accelerate the base calling or do bioinformatics, and it has fast SSD. And the specs of the tower scale with the number of flow cells that your box can run. So something we have been working on a lot is the robustness of the platform, and that comes from many areas. It comes from software and firmware, it comes from the chemistry, and it comes from the flow cells and chips. So with regards to software and firmware, some of the things we've been working on are things like better temperature control of the PromethION, and that will give us higher outputs, more reliable outputs, and it can also affect base calling accuracy. So that's a real key thing. Saturation detection, that's when a membrane isn't set up right and it pops under the stress of sequencing, and we have to turn that off. So that's been improved. We're probably a bit too twitchy actually, we're probably turning a few too many off. Electrocalibration, so that's the voltage, the applied potential you apply. We want that to be as tight as possible, because if that's tight, that gives you a really tight distribution of accuracies. We've also improved the flow cell check, so you might have experienced this recently. The latest software and firmware updates allow you to get a better representation of how many good channels you've got. So all of these things we work on do have small incremental improvements to the sequencing, so please do make sure that you keep your software and firmware updated. So I'm going to spend a few minutes to talk about flow cells. So stability of flow cells is absolutely key for us. We make the flow cells, we form on the membranes here in Oxford. We QC everything that goes out the door. And unfortunately, what we've had on PromethION is we've had a few problems with the flow cell quality. And this has meant that that although we catch a lot of these problems before they leave us, sometimes some of the more marginal cases will manifest when they're put under stress in sequencing, and that obviously affects your run. So we've been working really hard over the last 6 months or so to remove a lot of this variability. There's got a couple of pictures here. The first is where we've set the system up right. So this is a view on the top of the flow cell, view of the membranes, and I've marked them up in green circles. They're really nice and consistent. They're all the same size, and that will have a really good shelf life, and it will also have a really good sequencing performance because the chemistry is nice and homogeneous. That's set up correctly. On the right-hand side, this is what we see when some things go wrong. We can have a membrane that's too small, which is shown in the blue there. That might be a bit resistive, or it may not take a pour. We can have membranes that are slightly too big. They might be more prone to taking a second pour. They might be slightly leaky, or they might pop during your sequencing run. So we don't really want that, and we make a lot of the flow cells to the build on the left-hand side. But occasionally we do make flow cells that aren't of that quality, and you'll either see that in the flow cell check or in the sequencing run reports. So a good example is at the bottom here. We have very few kind of light blue and black fails, and even fewer of the kind of orangey fails, which represent the kind of higher current multiples. And there's a lot of sequencing output, so that's performing pretty well. And the one above it, unfortunately, has more flow cell fails, and you can see that really affects your flow cell performance. So what's causing this variation? There are two issues I want to point to. Both of them have been resolved now. The first took us a while to find, but when we did, it also took us a while to solve, and that is that on PromethION, because we build on top of the ASIC, and I should just say this is both these things have only affected PromethION. MinION MinION, GridION, and Flongle all are set up in a slightly different way, so these problems we never saw on those platforms. They wouldn't happen on them. In this PromethION defect, you can see we have an electrode in the bottom left-hand corner connecting through to the ASIC. That's a via through to the ASIC, and what we want is a really continuous metal stack. So we want to have a good connectivity all the way through that device. And we've got defects. These red arrows show that we've got a crack, and there's also a cavity. There's a void underneath that, and that is really not what you want in electronic product, because that will give you resistance or channel disconnects. And that's what we see in these chip maps. They come in all kind of shapes and sizes. White is a good kind of connected channel. Black is a disconnected channel. And we've got really good at detecting these. So most of these get caught in Oxford, but these kind of marginal cases might have crept through a few months ago. But thankfully now we've resolved the issue. We've changed the way that we put the metals down. We've improved the process, and we've also got a lot stricter quality control in place. So this won't be happening again. Knock-on effect is that that in changing that metal, we've also changed the lithography. And this took us a while to spot, but once we spotted it, thankfully it's very, very easy to change. So the wells are made using lithography. They're made by exposing the device to UV with a mask, and the UV dose kind of changes how good the structure you get out is. So you can see on the one marked up good, the structure is very well resolved, and with the green arrow you can see that those, what we call the fins, those are again well resolved, and that's really important because that acts, that has a capillary effect on all the fluids we add and holds them where we want them to be. That's what gives you your membrane stability. On the right hand side, this is where the change in the metal has caused it to be reflect the UV. This means that it's overexposed and those are clogged, and that means that structure just won't hold on to those fluids as well as we need it to. It will probably fail QC, but if it doesn't, it might fail in sequencing. Good news is it's a very easy thing for us to fix. Now we've spotted it, we can just readjust the UV dose and we can inspect. And now we're making things that have a really tightly controlled metal stack and have real tight lithography. So those are coming through in volume now. What happens when you take all those improvements and you put them together? How does it perform on a PromethION, which is really what matters. And we repeated our kind of box benchmark, and we took some of these flow cells, we just booked them out stores, we rolled in the latest Minnow updates, and we also used the kit with the fuel fix, which Stu and Rosemary will talk about as well. But what's important about that kit is it means that you can load up your box, you can put all these 48 flow cells onto your box, put your samples in, flush them through, and then press go and walk away. So this run, once it was started, didn't have any intervention. Our previous box run, our box record, we were at 7 terabases. This time we're at 8 terabases, and that's without intervention. So we were really happy with that. Another thing we were happy with was just how well the flow cells performed in terms of consistency. They were much more consistent than the last time we did this. They were running human samples, 48 flow cells, and we got a median of 170 gigabases, with the best flow cell on there being 200 gigabases. So really that consistency is what we're looking for, and we got both consistency and high output numbers. So we were very pleased with that. Another thing to look at is the accuracy. So we were hitting 96% modal raw read accuracy, and again what was really impressive was that was on every single flow cell. So it's looking good from our perspective, but this is an internal run, it's a benchmark, and in some respects that doesn't really matter. What really matters is how they perform with you. You're the ones who are using PromethION to solve really complicated biological questions, and I'm really looking forward to seeing how those flow cells perform in your hands over the next few weeks. And with that, I will pass over to Stuart Reed, our VP of Development.
Sorry for the delay, we're just wiping down the clicker. Nice and clean. Okay, so I'm going to talk to you about technology development, basically where we are and where we're going. But before I do that, I'd just like to talk to you a little bit about how we do things at Nanopore. From the very beginning, we've been community driven, which means that we release things early. We want feedback from the community because you guys know what you want to do with our product, how you want those features to develop, and we need that feedback in order that we can adjust what we do. And that means we can move very quickly. We have a very rapid cycle of innovation and development. But times change. We're not as young as we used to be. And now a lot more of our community are now over towards this right-hand side, the more mainstream users who just want things to work every time. They want robust, predictable products, stable supply. And James mentioned a lot of the things we're doing to enable that. So I'm going to start with a few slides on accuracy, and really when it comes to accuracy, what really matters ultimately is whether you get the answer you're looking for from your experiment. But you all do very different experiments, so we need to give you surrogate metrics. I'm going to talk through a few of those surrogate metrics from the raw read accuracy, consensus accuracy, variant calling, classification accuracy, those sorts of things. So So raw read accuracy, and James mentioned we're now at 97%, greater than 97% modal raw read accuracy, which is greater than Q15 on this log scale. And that's come from a development of our flip-flop neural network base caller. So we're talking about raw read accuracy here. So you have one signal, one piece of DNA going through a nanopore, and you base call that, and this is the accuracy that you get from that. And our production base caller, the flip-flop base caller, that processes that signal, and the flip-flop refers to, we've given the base caller a bit of a clue as to how to count. So it can count bases, it can count through homopolymers. And the change we've made recently is we've upped the stride. So it steps through the data more quickly in bigger steps, and that means the output is more information dense. It's also more computationally efficient, and that's let us grow the size of the networks a bit, and we can squeeze more accuracy out. So that's where that new accuracy has come from. And that's been released in Guppy 3.6 for about a month now, and that's just going into live base calling in Minnow this week. And as I say, we're at 97% modal. We also test on community data sets, so we're using this human data set from the Telomere to Telomere Consortium, and that's up there as well. And we've had some good feedback from the community re-base calling their old data. This is on R9 data. You can re-base call that data you've had lying around for the last few years, and you can now get these levels of accuracy. But we're not done there. That's our production base callers. We've also got two very active research programs running on base callers. One of them is the run length encoded base caller, and that gives the base caller a different clue about how to count bases. Rather than having to count them, it just tells you this base and this many. And we've also got the Bonito project, which is intended to be very simple, and that's as assumption free as we can make it. It's entirely based on convolutional networks, so there's none of the recurrence that makes the computational aspects hard to optimize. So we think we'll get quite a long way with Bonito, and those are both available on GitHub now for the early side of our community to have a play with if you like getting into code. Whenever we get a base caller model that's compatible with our production base callers, we're now going to put those in this Ririo project on GitHub. So you can download those models, add them to Guppy, and base call with our production software. And you'll also get modified base models and things like that that are compatible with some of our other research tools like Megalodon. And when you have a model that's compatible with our production base calling, you can reap the benefits that you get from our production software, particularly from Guppy. And one of those benefits is barcoding. So what is barcoding? So in this context, barcoding is a way of attaching a specific DNA sequence to your samples, and then you can mix those samples together, run them on the same flow cell, and then after the fact you can work out which sample is which from this DNA barcode. And our typical barcode is about 24 bases long. We don't really talk much about our barcoding, but a lot of people ask us how accurate are we? Well, we've rerun the benchmarks with our latest base cores, and we find that under our most stringent settings, you can now get to 99.999% classification accuracy. So our barcoding is pretty good. And if you're going to use more than one read to make any sort of decision based on your data, it's incredibly unlikely you're going to get that wrong, and you're probably dwarfed by anything that can happen upstream of our informatics. So on to the next form of accuracy, which is consensus accuracy. So consensus is where you add reads together to determine multiple reads to determine what the consensus sequence was. And the good news with nanopore data, you've got 97% raw read accuracy, but the most of that 3% that's left, the majority of that averages out when you add reads together. So most of that error is gone. With the latest, I should note though, that's when you use a tool that understands the data type. Our tool is called Medaka, so we really do recommend you have a look at our pipelines and tutorials that we've got available on GitHub and in Epitome Labs, and you can see how we do this, and hopefully you can achieve the same sorts of results that we get here. So with the latest base callers and the latest Medaka models, what we're finding is our long-term stable chemistry, the R941 chemistry, that's taken a big step forwards. That's now at Q45 accuracy for these bacterial genomes. That's fewer than 1 in 30,000 bases being in error. And our early access R10, which is just moving towards mainstream, that's now at Q50, which is fewer than 1 in 100,000 errors. So really high quality, fully resolved circular bacterial consensus genomes. But what if you want those levels of accuracy, consensus levels of accuracy, but for individual molecules? Well, you can do high single molecule accuracy with Nanopore. There's a couple of ways of doing that. One of them is with these things we call unique molecular indicators. And I mentioned with barcoding we're now pretty good at that. So you can exploit that here, and so you can put a semi-random barcode onto your DNA molecules. You can then amplify them, and then when you sequence them, you cluster the barcodes together, and you know which reads came from which original DNA molecule. And you can get up to consensus levels of accuracy for those individual DNA molecules. If you only want to do very sort of shallow single molecule accuracy, you can actually get it beyond Q20 with only 2 molecules using a feature of the Bonito research base caller, which is this pair decoding. So that simultaneously calls 2 signals at once. So you can get above Q20 single molecule just with 2x depth there. Now this plot up on the right here, and this is with the higher depth Medaka consensus single molecule consensus results. I've switched off all the filters. You got to be careful with other people's shots of genome browsers. There's various filters in there that have been built in for other sequencing technologies. We switch all those off here, so there's no quick consensus mode, there's no hiding of indels. You can see everything in that plot, and there are very, very few errors with single molecule sequencing there. It's not the only way you can do it. You can also do a sort of a depth-first consensus, if you like, and that's where you can put multiple copies of the sequence into an individual molecule. And perhaps the most mature way of doing that is from Cyclomics, who are a spin-out from the University of Utrecht. And they use this rolling circle method. And their example use case is the TP53 exon, which they use for detection of head and neck cancer, although you can apply this across a wide range of applications. And they really need to know the exact sequence of each molecule, because you're looking for a very small proportion of circulating free DNA molecules that have this mutation that's indicative of an issue. And they get up to consensus levels of accuracy with coverage, as you might expect. So very high single molecule accuracy with Nanopore data. Now for the next few slides, I'm going to talk about human whole genome sequencing. And you'll see on these benchmark numbers, you'll see I've used a 60x coverage number, which comes from 180 gigabases from a PromethION flow cell, which we think is a reasonable top-end expectation. Now we've resolved a lot of the issues that James mentioned earlier. So that's what we think you can expect from a PromethION flow cell. So when you're doing variant calling, as with consensus, it's important to use the right tool for the job. And again, we use our Medaka tool for this. And we've got a pipeline on GitHub, and there's an Epitome Labs tutorial, so you can see how all that works. And the way Medaka works is first, because you've got long reads, you can phase those SNPs, so you can work out which chromosome an individual read, which copy of the chromosome a read comes from. And then you can turn a complex heterozygous SNP calling problem into a much simpler homozygous SNP calling problem. And one of the changes we've made here is we've now got specific models for heterozygous SNP calling and homozygous SNP calling. And that improvement, coupled with the latest base callers, means we're very competitive in terms of single nucleotide variants with the short read benchmarks. So we're up above 99% accuracy there. We're a little bit behind on indels, but you can see the R10 chemistry really closes up that gap. Where long reads really matter, though, where they really add benefit, is with structural variants. And structural variants are defined as changes of more than 50 bases. And it's thought that structural variants account for perhaps 10 times more variation of bases between individuals than single nucleotide variants. So all sorts of things happen with a structural variant. Things can be swapped about, they can be deleted, copied, and even swapped over from one chromosome to another. So the way you analyze this is you align reads to the reference, and there's a particular tool called Sniffles you can use to call variants. That was a community developed tool. We've got a workflow for this in Epitome, point and click, simple as it can be. We've got a GitHub pipeline if you want to integrate into your own systems, and we've got Epitome Labs tutorials if you want to learn a little bit more about how that works. So evaluating against the benchmarks, using this 60x coverage number, we're up at 96% accuracy there, which is as good as anybody else out there. But I've got this 15x line on the table, and we're still getting reasonable results there, and we think there's a bit of scope in the algorithms to improve these things. So if you're happy with results at 15x, you're now talking about being able to do 4 human genomes on a PromethION flow cell at that 180 gigabase number. So another thing long reads are good at is assembly, and this is an area we're just beginning to get into. The community are way ahead of us here with tools like Shasta from the team at UCSC and the Chan Zuckerberg Institute. So what we've done is we've taken our research run length encoding base caller, and we've applied that, and done assembly with Fly and Shasta, and then Medaka polishing, and we've looked at R9 data sets, we've looked at R10 data sets, and we've also looked at the telomere to telomere data set, which is from CHM13, which is a homozygous cell line. And we get really pretty good contiguities. It's fashionable in this field at the moment, it appears, to measure accuracy by a reference-free K-mer comparison method. That's called Mercury, so we've done that, and we see the accuracies are pretty good too. And the Busco scores are basically complete. And you can see a picture of the assemblies there, where they're marked up by breakpoints. You can see you've basically got complete chromosome arms assembled there with nanopore data. And we're talking volumes of data that you can get from a single nanopore flow cell to get those sorts of assemblies. And so that means you get your SNPs, your structural variants, and your assemblies all from the same flow cell of data. But nanopore data is unique in that you're looking directly at the DNA, and you can see those modifications. And James mentioned a little bit of this earlier. So you can directly measure modification. And because I'm talking about human sequencing, I'm going to talk about 5-methyl-C in this CPG context. We've previously spoke about all context models for perhaps for bacterial or plant modifications with 5-methyl-C and 6-methyl-A. We can do that. I'm just going to talk about the human context for now. And what we see is from the same data as we've used earlier, when we compare our 5-methyl-C calls to the ground truth out there, which the best there is this bisulfite sequencing, which is a chemical conversion. You then put into your short read sequencer, and you have to do a sequencer converted sample, a non-converted sample, and compare them and find out where the changes were. That indicates where the methylation was. We find that the nanopore data is better correlated to this bisulfite sequencing data than a bisulfite sequencing run is correlated to another bisulfite sequencing run. So we think we're closer to ground truth than bisulfite sequencing when it comes to 5-methyl-C. But it's not just 5-methyl-C you can see, it's all in there in the signal. So we can also teach our base callers to see 5-hydroxymethyl-C. So we've now got a model that's trained for both 5-methyl-C and 5-hydroxymethyl-C, as well as the other 4 bases. And we apply that again to the same data. And you can see that the signal in the aggregated plots there, the 5-hydroxymethyl-C signal is associated with transcript start sites. And as we understand it, that's consistent with the biology there. So you've got your SNPs, your SVs, your assemblies, and now your base modifications, all from the same data, perhaps from one PromethION flow cell. Okay, so that's accuracy. I'm going to move on to talk a little bit about software. We've got a new UI coming. That's coming with Minnow 4, which is imminent. Those of you with a Mark 1C will be familiar with this UI. It's been redesigned from the ground up, largely with a touchscreen in mind. But there's also a lot of personalization you can do there. You can choose which graphs you want to see and which order you want to see them in. We've got Android and iPhone app versions in development. So if you wanted to manage a fleet of PromethIONs, 10 PromethION devices in your lab from your iPhone, we think you'll be able to do that in the near future. It's not just a new UI though. There's loads of good new features in there as well. In particular, we've got live alignment in there now. So now you can run sequence alignment from our UI, and it's all in there, included on your box. You're just simple as selecting a reference and optional bed file targets, and then you can see where you're getting your data in that run. And you can combine that with barcoding. So you can see for each sample in a flow cell, for each target you're interested in, have you got enough data, and you can run until you do. And that's also an enabler for read until, which I'll talk about in a minute. We've also got tutorials and sort of guided tours going into the UI, so it's intuitive and easy to use. And we're doing a lot of work on system management, things like that, which makes it easy for this mainstream user managing devices, just want it to work every time, seamless updates, that sort of thing. Behind the scenes, we've made big changes to our API, so that really lowers the barrier for integration with LIMS. There's loads of examples there. These APIs are there, open source on GitHub. We've also we've got planned changes coming to our simplest possible LIMS integration, which is a sample sheet upload. We're going to let you identify samples by barcode as well, so then that will then get written out into the data that comes out, and that will really simplify downstream processing and prevent sample mix-ups, things like that, when you've got the sample identifier written into your data. We're also switching on our compression by default now to help with file management. Okay, so read until. I said I was going to talk about this. It's got a new name, adaptive sampling. What you can do is, as that molecule is going through the nanopore, you can read out the first bit of sequence, and you can decide whether you want to continue sequencing that molecule or not. And if you don't, you can kick it out. And that lets you enrich for targets of interest or deplete things that you don't want. And you've been able to do this for a couple of years now. Earlier this year, though, we moved it from behind our developer license out onto general release. But it's, you still need to get your hands dirty in the code really to run this. Despite some great work from the community making tool sets. So we've got an integrated solution coming. It's going to be integrated in the UI. Perhaps not this version of Minnow, probably the one after. That's going to run on your Gridiron, your Mark 1C, and we're working on making it run for Methane as well. That's going to be as simple as selecting your reference sequence and your targets, and then the box will do everything for you. So that's adaptive sampling. I said you've been able to do this for a while, and there's been some great work from the community, in particular Matt Luce's group and Mike Schatz's group, with two different tool sets for doing read until, and they've done cancer panels and exome panels respectively. And our apps team have also been doing some interesting examples using Matt's toolkit that happens. But you can do this right now if you can't wait, but you do have to get your hands a little bit dirty. So it's not just the sequencing software. We also offer a range of tools for bioinformatics for downstream analysis. The simplest of those is Epitome. It's a website. It's point and click. It's real time, end to end. Upload your data, intuitive GUI, loads of workflows there, and you get your report back at the end. That's all cloud based. You don't have to do any management of compute. We do all that for you. That's your simplest solution. If you want to learn a little bit more about what's going on, perhaps tweak parameters, play with our data sets or your own data sets, then we've got Epitome Labs, and that's where we're going to deliver all our tutorials. And that comes as a pre-installed environment. And one of the headaches of bioinformatics is getting all the versions to match and everything installed correctly. That all comes pre-installed with Epitome Labs, and that runs on your device. If you really want to get your hands dirty, like tapping away at the keyboard, then we've got our open source GitHub projects, and I mentioned that's where all the research-based callers are, and that's where you can get hold of all our pipelines. It's all open source, customizable, integrate with your own platforms, and that's where the Minnow APIs are as well. So check that out if that's your thing. Okay, I'm going to move on to platform development. When we talk about the platform, James has covered loads of stuff about what we're doing for robustness, really addressing that, make it work every time. I'm going to talk a little bit more about the futures of how much data you can get out of the device, and also touch on what you have to put in. So in terms of consistency and ease of use, those of you who've been doing perhaps short read sequencing or trying to get really high outputs are used to knowing about perhaps this speed drop-off. And that's where the enzyme consumes fuel even when it's not sequencing. And that means that to get the maximum outputs, you have to interact with the run and perhaps top up with fuel or top up with new library. And that's all going to be gone now. We've fixed that. That's coming out in the next month or so with Kit 10. That's where it's coming out first. And this will make it particularly easy for those doing short read libraries where this was a particular issue. But that's going to be gone. No more interaction with the run, no more topping up. Now, in terms of increasing output, we've got a couple of ways we can do that. One of the main limitations on how long we can run the device for is the electrochemistry in the well. And we've got two ways of addressing that. One, we can tweak the ratios and the concentrations and the exact chemistry that's in there. And we think by doing that, we can perhaps double the runtime. We've got proof of concept on that, so that's something we're working on right now. The other thing, it's like a battery, so you can make a better battery, or you can just use twice the number of batteries, so we can make the wells bigger. And what we've done there is we've actually brought some future developments from our next generation devices that we're looking at now. We're folding those back into our current devices, and we think we can make deeper wells and again double the runtime that way. So we think those two things will multiply together, so potentially 4 times the runtime. Those things are in development right now, so watch this space. I mentioned I was going to talk about what you have to put into the device, and James told you earlier that we achieve our sensitivity, where you get DNA down to the membrane with this tethering chemistry. But the tethering's messy. It doesn't just stick to the membrane; it sticks everywhere. And we think if we can get it to just stick to the membrane, then there's potentially a 200-fold improvement there. And we've got proof of concept on this. You can see the image in the middle there, which is we've got the DNA stained, and you can see the DNA is all right there on the membrane, and there isn't much anywhere else. So we're working on this at the moment. Again, currently in development. We've got about a factor of 10 improvement in sensitivity, but we're really aiming for 200x. And if you can get to 200x, we're talking a picogram, perhaps even femtogram levels of sensitivity. So you really don't have to put much into these devices to get a decent amount of sequence data out. So we're going to look a little bit further into the future now. I mentioned we'd pulled back a few features from here to make the wells deeper. We're going to look what we're doing slightly longer term. So this slide's really just a reminder. One of our core components of our platform is the ASIC, and we do have a new ASIC. We're still working on this. It's much smaller than both the MinION and the PromethION ASIC, and it's much lower power, and that opens up new formats like Plongle and the SmidgION mobile sequencing. So we're still working on this. Again, watch this space. One of the key limitations, as I said, was the well, and what limits the number of wells that you can get. Sorry, the number of channels you can get packed into a device is the size of that well. And we think that by changing the way we measure the nanopore signal, we can go from the current approximately 200 micron pitch down to about a 20 micron pitch. So that's a big improvement in packing density. And we do that by changing from a current-based measurement to a voltage-based measurement. So we're using a voltage divider to measure the same property of the nanopore, but in a different way. And we think that on a MinION-like device footprint, you can really pack those channels in, and we think that will get you to about 4 terabases a day on a MinION-like footprint of device. This is future stuff, but a few critical things we need to tick off to prove that we can do that. And these are the two things. We've proven that you can get 20 micron membrane pitches, you can form the membranes, and you can get nanopores in. So that's ticked off. The other thing we need to tick off was, can you get a decent looking squiggle, decent looking sequencing signal from voltage sensing? Yes, you can. We've done that. So again, this is in development. We've got ASICs designed to do these measurements, and we expect to have a play with those later this year. Okay, with that, I'll hand you back to the present with Rosemary to talk about our products.
Thanks very much, Stu. So in this section, what I'm going to talk to you about is how we take all of our lovely platform developments and the things that our teams are working in R&D and engineering, and how we package those up and get them ready for release. So the first thing we have to do is actually sort of group our products. Are they products for preparation, sequencing, analysis? But also we have to consider all of the different types of users that we have. So we have some of you who are taking the sequencers out into the Arctic, some taking it to the jungle, others working in your labs, some core providers, PopGen. All of these different use cases are now adopting nanopore sensing. And we have to work with you guys, and we like to work with you guys to make sure that we're establishing a really, really easy end-to-end process. So the first thing we have to do is prepare sample. as Stu and JC have mentioned before, you can actually sequence pretty much anything on the platform. You can get modified bases, structural variants. You might want to do get things for rapid time to results. And as such, the applications group and the development group at Oxford Nanopore have devised and developed either protocols or kits that can take native DNA, amplified DNA, RNA, either through cDNA or direct, and also targeted methods. And we provide a whole host of kits available on the shop for users to be able to apply to their research. Over the last few months, we've been working with our production scale customers to start increasing the number of genomes that those guys are capable of sequencing each and every day. So we've had XL kits coming out. We are actively working on automation with Hamilton, with ligation kits, native barcoding, the Arctic COVID prep, Lampore, that's all going into the automation prep. And also some of the upstream processes that you have to do to your sample in order to feed it into these automated preps. All of that is being worked on in development to help people run more smoothly, have a better experience at the high end of sequencing. With improved output, also comes the ability to sequence more samples on a flow cell. And many of you have asked for a long time for more barcodes. So this year, you're going to get more barcodes. There are going to be 96 rapid barcodes. Those are in development. The 96 native barcodes are on order. We are planning to ship those in the next couple of weeks. And dual barcoding is already as a protocol on the community. So barcodes and barcodes galore. Stu's mentioned how accurately we can call them, and we will keep making sure that we fulfill the need of users to barcode their samples. Some of you also have your own barcodes, and as such, over the next day or so, we will be releasing the information on how to do custom barcoding in Guppy, so that for those who've already developed barcodes for your own workflows, you can just apply Guppy to deconvolute your samples. Once we also cater for all of those users who want to automate things on a lower scale. So let's think about Voltrax. This is a program that has been very active at Oxford Nanopore for the past couple of years. It is a product that is designed for users to load their samples, start a protocol, and the device takes care of all of that in software. So you have a small device, you've got a cartridge, and on the device you've got fluorescence detectors, magnetic elements, Peltier's heaters. So you should be able to do pretty much everything you need to do in a complex experiment. And what happened at the start of this year was this rather incredible workflow had to be devised and developed, and was devised and developed by the Arctic team. And it really provided a very good challenge to the Voltrax team at Oxford Nanopore to say, how are we going to automate something as complex and as involved as an Arctic prep onto our Voltrax prep device. So we're showing here the video that we've been showing is actually the Arctic prep running on Voltrax. It does an RT, it does a PCR, it cleans up, it does a QC. Then we add our rapid barcodes, rapid adapters, and we get the resulting library ready to sequence. And the whole premise for the team was really to use this to exercise all of those last features that are really, really missing for the Voltrax community to start really running this in anger. So I'll talk to you a little bit about where we are with those features. So the first one is PCR. So that is very active in active development internally. We've got the software and the chemistry ready. What we found is that we get great results internally, but we want to make an upgrade to the cartridge in order for that performance to be robust in field. So our engineering teams are currently working on that. On the fluorescence, again, we've got our software and hardware now working, and we'll be rolling that ability to detect your DNA or RNA over the coming weeks and months in new scripts that we'll be pushing out into the field. On our software has actually been hugely worked on in the background. We have a new UI, but there's actually behind the scenes a lot of work has been going on. So the new UI is launching now. You get live cartridge feedback. With the rewriting of software, what we believe is that we can get to a cross-platform by the end of 2020. And we know that one of the key questions that we get from the Voltrax community is, can I run it on my Mac? And soon the answer will be yes. The whole point and premise of Voltrax and the power behind it is to enable users to program their own sample prep onto the device, and we will be opening up an API to developers over the summer so that they can start experimenting with this, giving us their feedback so that we can take this feedback into the development of the final features and software. And finally on Voltrax, despite the whole premise of the product is one that is easy to use, but right now you need to pipette in something like 15 different reagents. And so what our engineering teams are working on internally are these reagent loading cassettes that you can simply slot into your Voltrax cartridge, press, and the reagents get dispensed. So that should really reduce the need to pipette on a product that is designed to be simple. So now that we've covered a lot of the sample prep options, we're going to take you through sequencing. So we have a very, very scalable offering. James and Stu have already mentioned this. So we've got flongals at 1 to 2 gigs, MinIONs, MinION 20 to 40, GridION multiply that by 5, and then the PromethION, you get that 100 to 200 gigs per flow cell and the 8 terabase run. And on the right here, I have actually I have a chart that was designed by Clive and Spike back in 2010, and it goes to show really still how relevant this chart still is, because you can massively increase the output of your device simply by increasing speed, increasing runtime. For those of you who started with us back in 2014, you might remember that we were processing, our enzyme was processing DNA through the pore at 30 bases a second, and then we sped that up to 200, we're now at 400, and so we're getting to those outputs of 20, 30 gigs on a MinION flow cell. You multiply that by 5, you get a GridION, but actually in that same spot, a GridION, you also have a single PromethION flow cell. So it's always interesting when you devise these plots, do you show the GridION or a PromethION cell in there? And of course the PromethION is capable of achieving much more. So with the developments that we have in R&D coming with the deeper wells, with the different chemistry, hopefully extending those runtimes, it might mean that you can actually get towards a human genome on a MinION at some point in time. So just to reiterate key upgrades on the output that are shipping, Kit 10 will be installed during July, ships in August, XL versions will follow. You don't need to top up anymore for short read libraries, things like cDNA. You can go from sort of 100 million to 160 million reads on your PromethION flow cell. And then on the PromethION, on the updated flow cells that gave our teams here 8 terabases, we will be communicating with all of the PromethION accounts. We will be shipping them some latest flow cells with some Kit 10 to hopefully welcome some of you back to work. We know many of you have been home for the last few weeks. On system performance, on accuracy, we've got that high modal raw read accuracy, SNP detection, SV detection, assembly, methylation included, and always the key drivers are our algorithms, our chemistry releases, and all of the pipelines that the community work really, really hard to develop. And so the key, some key updates that we've had recently, of course, are the R10.3 nanopore. So this is the nanopore that improves our homopolymer resolution and delivers that higher consensus. And it's been released on MinION since January in shipping. And it is now with early access PromethION users, with Keygene having run it last week, I believe. And Matt Luce, of course, waking up early this morning to put some runs on. So thank you for trying it. And we look forward to your feedback. On the algorithm side, of course, we've got the GitHub releases that we did earlier this year in March for both the flip-flop but also the Bonito code. In May, we put those in Guppy, and that Guppy release will be getting into Minnow in the next week during June, so that all of you can start having the benefit of that improvement in accuracy over the next few days. We also, I believe, Chris Seymour's actually just done a Bonito upgrade as well, so more things coming from his project. As well as looking at the accuracy of our algorithms, we always have to keep in mind the speed. And with that, we work incredibly well with partners such as NVIDIA. And earlier this month, we've enabled users who have their laptops, we've enabled them, if they've got a Linux laptop, to plug into the GPU that they have there to accelerate their analysis. But also we then have all of the cards that are embedded inside our products, like the TX2s and the GV100s. And we're very excited by things like the A100s and Jetson NX for future products. We then move on to our analysis product. So mentioned already by Stu is our Epitome Labs, which is something which is a mechanism by which users can get to know how to run the analysis pipelines. In there, we've got our QC, small genome assembly, SV detection, metagenomic analysis, the base mods, the SNPs, and in development, there are a whole ream of analysis pipelines coming, things such as transcriptome exploration and clone validation. The Epitome group have also been incredibly busy for the last few months. Of course, they have to deliver very, very clean, very simple to use workflows. But they also have been working incredibly hard on their agent. So a few new features, if you go and talk to them in the live lounge, they'll walk you through the new agent, which is in beta testing now, and they are looking for people to join that beta testing group. But you will see features like previous runs, multiple upload data sources, live summaries, and multi-user support. So we're very, very excited with the latest Epitome Labs agent coming through. So now that we've covered the pipeline of products from preparation to sequence to analysis, I'll just take you through what are our core values when we decide to launch a product or bring a product to market. And there are really four. The first thing is, because we want to democratize sequencing, it has to be an accessible technology. It has to be capital free. We want transparent pricing. We want everyone to understand what they get out of the technology. We also are a company that have very a strong track record of upgrading our technology. So the upgrade paths have to be simple and easy. And of course, we are really based on community feedback that goes into our future product development. So on our capital free, on that accessibility, our starter packs and our devices can all be accessed with consumable budgets. There are capital versions as well, but consumable budgets are really where we all started, with a starter packs in the MinION at only $1,000. And all of those now include that 12-month warranty, and they all include kits and flow cells as well. So that goes for our capital free model. With our transparent pricing, we have all of the pricing of our flow cells online, and if you think about sort of current outputs, future outputs, we are going to be delivering incredibly competitive price per gigabases and enabling people to do larger and larger projects without big capital investments or without huge investments in consumables either. So it just really tries to bring biological information to the masses. With more multiplexing, with more barcoding, those of you who are doing smaller genomes, targeted work, can now access really low and very competitive price per samples and get through hopefully a lot more of your samples, a lot more of your projects than you would have done previously. And when you apply those kind of models to what users are using our technology for, be it whole genome sequencing, targeted RNA metagenomic, you can come up with a few very basic user cases. But for example, in human genetics, some people are doing human genetics on MinION GridION. Most people are running it on PromethION. Currently, with our lowest price per flow cell at under $1,000 per human genome, as soon as we get that chemistry out and those improvements out that get you towards the 200 gigs, you'll be able to be running 2 genomes per flow cell, bringing that into well under $500 a genome. So we really have a very competitive offering on whole genome sequencing. With regards to small genomes, we've really had a huge amount of projects on smaller genomes for years and years. And of course, with more barcodes comes that ability to run more. But really, people pick up our MinIONs and our devices for smaller genomes, really because they can get started for as little as $1,000. They take their research to their samples, and they get their answers really quickly. On targeted sequencing, we now have 3 options. We have the amplification-based, and so here we have rapid PCR methods. You can do high multiplexing. Now with all of the UMIs, the RCAs, you can do rare variant detection with incredibly clean and high accuracy data. And we can also do amplification-free targeting. So the Cas9 kit is available in store. And there you can retain all of your methylation information. You can capture reads up to 100 kbs, do complex variants, and all of the things that you would want to do on a whole genome scale, but perhaps you want to sort of save yourself some capacity or multiplex. Then we move on to sample prep free. This is, I think, what everyone is really excited about. This is the adaptive sampling, where you simply add your target to the software, you put your library inside, and you walk away, and the system does it for you. So I think this is particularly one thing that we are incredibly excited about, is what are all of the crazy applications that you guys are going to come up with regards to adaptive sampling. So we look forward to hearing all about those over the summer. On RNA sequencing, there have already been some really great sessions earlier today at the conference. Of course, with cDNA, you get your long reads. They span the whole transcript. But also with the improvements with Kit 10, you get millions of reads. So you can do isoform and expression studies in one flow cell. And of course, with direct RNA, there is no other way to go off and find all of the methylation information and all of this really, really great biology that is coming out of all of the direct RNA societies and speakers that we see. Then finally, metagenomics. This one it's really hard to pick a favorite project, because these projects, there are so many projects, so many applications, so many great stories that have come out of the metagenomic community. I've just picked 3. One of them is that metagenomic assembly, using that long read to get complete assemblies out of mixed samples. And then I've also picked on the River Metagenomic publication that came out earlier this week, really actually because it's the Mark group. And for those of you who were in London Calling 2015, the Mark group was a sort of collaboration between kindred spirits of the nanopore community. And they all came together, first of all, to understand if someone sequenced an organism in New Zealand and someone sequenced that same organism in Canada, would the answer be the same? So they've made quite a lot of different projects since then, but this is the outcome of, I know, a really, really long collaboration, and what they've done is sequenced 11 rivers across 3 continents and been comparing the microbiomes of those rivers. More recently, of course, there's been a really big push on metagenomics in the fight against COVID, with lots of users deploying the technology for its speed, ease of use to detect co-infection in COVID patients. So then back to the final 2 values, that upgrade cycle and that community-based feedback into product development. So we thought it'd be fun to sort of show you a little map of all of the user journeys that we've been on. So for those of you who started with us in 2014, you got your map MinION that got upgraded to a Mark 1 in 2015, a Mark 1B in 2016, and all the way through that, we have always provided users with a very, very simple and easy upgrade path. But also, actually, it's important to note that most of the upgrades actually happen in the consumables and the software and the kits. So someone who bought a Mark 1B MinION in 2016 will have been getting 2 or 3 gigabases of data, and today on that same device, they'll be getting 20 to 30 gigabases of data. So the upgrade path, really, the majority of it happens behind the scenes, happens inside the flow cells, inside the kits with software downloads, and we experience this growth of performance together. On the community-driven feedback, we love our developer success stories. We really actually thrive on getting product out of the door, out of R&D, really very, very quickly. And that's because when we try something internally, we could spend 10 months getting it perfect and then ship it, and we've forgotten a killer feature. So this is why it's really important to us to be connecting with users very, very early, and anyone can be involved. We do have a feature request pin board on our community, and you can ask for new features for software, but also, you know, how do you like to buy things? How do you like to store things? There are actually all sorts of topics up on that feature request pin board, so we do really welcome that input. And so what we've seen over the last few years is this sort of growth of the community, this evolution of the community from those very early access users that started with us in 2014 to people then doing metagenomic studies in 2016, 2017. PopGen started to come at us really thick and strong with the birth of PromethION. And we also have a lot of people in translational, in clinical, and applied markets. And what we thought we would start doing, and what you will start to see and experience in our shop, is that we're going to start really giving all of you guys a much clearer roadmap and a much clearer picture of where all of our products are. So you'll have early access shops where you can get your hands on all the latest innovation. You'll have the research use store where you can keep buying all of the products and all of the devices that you're using today. Of course, we've got a Q-Line and a Lampore store to come as well, and Gordon will be talking to you about Q-Line tomorrow morning. So we hope that by providing everyone with a really clear guidance of where things are, we're going to be better managing everyone's expectations. And what we really want to do is really carry on innovating whilst providing all of the users who really want a robust, easy to use experience, providing them a really clear place to go. So then comes the homework slide, everyone's favorite. So this is a summary of all of the releases and all of the developments that we've talked about today. And actually, for the first time, we're going to discontinue something as well. So we have on our device pipeline, we are, GridION Q is coming, GridION Lampore is coming, the Mark 1C is shipping globally soon. And as soon as that starts, then we will be having a discontinuation program for a minute. But I think everyone, we might have talked about that before. We have got a Voltrax V2 cartridge upgrade coming as well later in October. On our flow cells, PromethION R103 is now in early access, and we will be having early access of low input flow cells and long life flow cells later in the year. The kits are too many to mention in one go, and the software, of course, Minnow4, Epitome new agents are going live sort of very imminently, and alongside a lot of Guppy updates. So with that comes to a close the technology update, but also a reminder that there is a huge amount more coming, and Clive will be doing a broadcast during the summer. But with that, we will welcome Stuart and James back onto the stage, and we will take some questions.